Peptides. The importance of peptides. Peptides participate in a number of biological activities. They serve as intermediates in the formation of proteins. They appear as constituents in group compounds called alkaloids. Majority of these have been isolated from fungi, although they are also found in higher plants. Ergotamine is the peptide alkaloid from ray ergot and has pronounced pharmacological properties. The four components of these alkaloids are lysergic acid, alanine, proline and phenylalanine. Many of them possess antibacterial activities and are usually present in fungi and bacteria. Penicillin G with three components, valine, cysteine and phenylacetic acid, is a common antibiotics. Certain other peptides serve as growth factors. Folic acid, a water-soluble vitamin, is a noteworthy example of it. Another group of peptides serving as growth factor for a variety of microorganisms is streptogenin. Higher animals do synthesize certain peptides serving as hormones. Certain peptides like glutathione participate in controlling the oxidation reduction potential of the cell. This also serves as a key intermediate in electron transport systems. A direct correlation has been found to exist between the amount of peptides in the urine of patients and their mental state of disturbance. The peptides have been shown to introduce in animals some of the conditions for the development of psychotic disorders, which leads to mania, depression or schizophrenia. The urine test can thus indicate if the person is suffering from mental illness. The case of this hypersecretion of peptides is not yet well established. It may exist from birth in organic genetic rearrangements or may be introduced in healthy people by environmental factors. Peptide hormones. Peptide hormones or protein hormones are hormones whose molecules are peptide or proteins, respectively. The latter have longer amino acid chain lengths than the former. These hormones have an effect on the endocrine system of animals, including humans. Most hormones can be classified as either amino acid based hormones, that is amine, peptide or proteins, or steroid hormones. Examples for peptide hormones: gastrin, glucagon, growth hormone, insulin, oxytocin, prolactin, vasopressin, etc. So we will see about some examples. Vasopressin. Vasopressin, also called antidiuretic hormone, that is ADH அப்படின்னு சொல்லுவாங்க. Original vasopressin, AVP or argipressin அப்படின்னு சொல்லுவாங்க, is a hormone synthesized as a peptide prohormone in neurons in the hypothalamus and is converted to AVP. The vasopressins are peptide consisting of 9 amino acids, that is nanopeptides. Vasopressin, also called antidiuretic hormone that plays a key role in maintaining osmolality, that is the concentration of dissolved particles such as salts and glucose in the serum, and therefore in maintaining the volume of water in the extracellular fluid, the fluid space that surrounds the cells. Next, normal serum osmolality ranges from about 285 to 300 milliosmoles per kilogram in healthy people. Vasopressin is used to treat diabetes insipidus related to low levels of antidiuretic hormone. So next example is oxytocin. Oxytocin is a peptide of 9 amino acids, that is nanopeptide. The structure of oxytocin is very similar to that vasopressin. Both are nanopeptides with a single disulfide bridges, differing only by 2 substituents in the amino acid sequence. Difference from oxytocin or the phenylalanine and arginine. Oxytocin is a peptide hormone and a neuropeptide. It is normally produced in the hypothalamus and released by posterior pituitary. It plays a role in social binding, sexual reproduction, childbirth and the period of after childbirth. Oxytocin is released into the bloodstream as a hormone in response to the stretching of the cervix and uterus during the labor pain and with stimulation of the nipples from breastfeeding mothers. This helps with birth, bonding with the baby and milk production. Due to its similarity to vasopressin, it can reduce the excretion of urine slightly, and so it can be classified as antidiuretic. In several species, oxytocin can stimulate sodium excretion from the kidneys, that is natriuresis, and in humans, high doses can result in low sodium levels, that is hyponatremia. The next topic is peptidases. A protease, also called as peptidase or proteinase, is an enzyme that catalyzes proteolysis, the breakdown of proteins into smaller polypeptides or single amino acids. They do this by cleaving the peptide bond within the proteins by hydrolysis. A reaction where water breaks bond, proteases are involved in many biological functions, including digestion of ingested proteins, protein catabolism, that is breakdown of proteins, and cell signaling. Without additional helping mechanisms, proteolysis would be very slow. Taking hundreds of years, proteases can be found in all forms of life and viruses. They have independently evolved multiple times, and different classes of proteases can be perform the same reaction by completely different catalytic mechanisms. There are two types. One is endopeptidases, another one is exopeptidases. Now coming to the endopeptidases. Endopeptidase or endoproteinase or proteolytic peptidases that breaks peptide bonds of non-terminal amino acids, that is within the molecule, in contrast to exopeptidases, which breaks peptide bonds from n piece of terminal amino acids. For this reason, endopeptidases cannot break down peptides into monomers, while exopeptidases can break down proteins into monomers. A particular case of endopeptidases is the oligopeptidase, whose substrates are oligopeptidases instead of proteins. They are usually very specific for certain amino acids. Examples of endopeptidases, that is trypsin, cuts after arginine or lysine unless followed by proline. It's very strict, works best at pH 8. Chymotrypsin cuts after phenylalanine and tryptophan or tyrosine unless followed by proline. Cuts more slowly after histidine, methionine or leucine. Works best pH at 8. Elastase cut after alanine, glycine, serine or valine unless followed by proline. Thermolysin cuts before the isoleucine, methionine, phenylalanine, tryptophan, tyrosine or valine unless preceded by proline. Sometimes cuts after alanine, asparagine, histidine or threonine. It is each stable. Next, pepsin cuts before leucine, phenylalanine, tryptophan or tyrosine unless preceded by proline. Also others quite non-specific work at pH 2. Glutamyl endopeptidases cuts after glutamine, works best at pH 8. Exopeptidases. An exopeptidase is any peptidase that catalyzes the cleavage of the terminal or penultimate peptide bond. The process releases a single amino acid or peptide form the peptide chain. Depending on whether the amino acid is released from the amino or carboxylic terminal, an exopeptidase is further classified as an aminopeptidase or a carboxypeptidase respectively. Thus, aminopeptidase as an enzyme is a breast border of the small intestine which cleave a single amino acid from the amino acid terminal, whereas carboxypeptidase, which is digestive system present in the pancreatic juice, will cleave a single amino acid from the carboxylic end of the peptide. Biologically important peptides. Several peptides occur in a living organisms that display a wide spectrum of biological functions. Generally, the term peptide is applied when the number of constituent amino acids is less than 10. Some examples of biologically active peptides and their functions are described here. First one is glutathione. Glutathione, it is a tripeptide composed of 3 amino acids. Chemically, glutathione is glutamyl cystinyl glycine. It is widely distributed in nature and exist in reduced or oxidized form. Functions is, in a steady state, the cells generally maintain a ratio of about 100/1 of GSS to GSSG. The reversible oxidation reaction of glutathione is important for many of its biological functions. Glutathione serves as a coenzyme for certain enzymes, example prostaglandin synthetase and glyoxylase. It prevents the oxidation of sulfidyl groups of several proteins to disulfide groups. This is essential for protein functioning, including that of enzymes. It is believed that glutathione in association with glutathione reductase participate in the formation of correct disulfide bonds in several proteins. Glutathione forms specialized function in erythrocytes. It maintains RBC membrane structure and integrity. It protects hemoglobin from getting oxidized by agents such as H2O2. Glutathione is involved in the transport of amino acids in the intestine and kidney tubules via glutamyl cyclormeister cycle. Glutathione is involved in the detoxification process. The toxic substances, that is organophosphatase, nitro compounds, are converted into mercaptic acid. Toxic amounts of peptidase and free radicals produced in the cells are scavenged by glutathione peroxidase, that is selenium containing enzyme. The second one is tryptophan releasing hormone. It is a tripeptide secreted by hypothalamus or TRH, that is TRH. Stimulates pituitary gland to release thyrotropic hormone. Next one is oxytocin. It is a hormone secreted by posterior pituitary gland and contains 9 amino acids, that is nanopeptide. Oxytocin causes contraction of uterus. Next one is vasopressin, that is antidiuretic hormone or ADH. The ADH is also a nanopeptide produced by posterior pituitary gland. It stimulates kidneys to retain water and thus increase the blood pressure. Fifth one is angiotensin. Angiotensin 1 is the decapeptide, that is decapeptide, 10 amino acids, which is converted into angiotensin 2, that is 8 amino acids containing peptide, octopeptide. The latter has more hypertensive effect. Angiotensin 2 also stimulates the release of aldosterone from adrenal glands. Next one is methionine encapsulin. It is a pentapeptide found in the brain and has optial-like function. It inhibits the sense of pain. Bradykinin and gliadin. They are nano, that is non and decapeptide respectively. Bradykinin is a nanopeptide, gliadin is a decapeptide. Both of them act as a powerful vasodilators. They are produced from plasma proteins by snake venom enzymes. Eighth one is peptide antibiotics. The antibiotics such as gramicidin, bacitracin, thyrocidin and antinomycin are peptide in nature. Next one is aspartame. It is a dipeptide aspartyl phenylalanine methyl ester produced by combination of aspartic acid and phenylalanine. Aspartame is about 200 times sweeter than the sucrose and is used low calorie artificial sweetener in soft drinks industry. Next one is gastrointestinal hormones. Gastrin, secretion, etc. are the gastrointestinal peptides which serves as hormones.